Hello and welcome to another video lecture from MrWatkins.com. Today's video is on the cell cycle. Specifically, we're going to focus on mitosis. I am a little hoarse today, and so hopefully I needed to get this done for my classes tomorrow, so hopefully you'll be able to understand what's going on. This was intended to be after you have had a chance to look through the notes that are on cell cycle. This handout is also available from me, so please make sure you stop by and pick that up. Remember, the cell cycle has these three pieces here. Really, for advanced bio, we say that it's interphase and mitosis, and that cytokinesis is part of that. There is some differences depending upon what program you're in as to whether that's true. But we're going to walk through the process so that you can fill in this graphic organizer. So interphase, and this is all meant to be colored. I'm not going to go through that process. You can go back in and color back over that. In fact, it's a good way of reviewing and maybe even getting to summary of your notes. So interphase is the first part of the cell cycle, and remember, the cell cycle is a piece that basically the life of a cell. So a cell is born, if you will. Cell divides and a new cell is formed, and that cell then immediately goes into interphase. And interphase has three parts, and three parts of that interphase are G1, S, and G2. Now these three parts are important because they set up that process of growth and reproduction that will eventually happen when we hit mitosis. Many cells stay in interphase for a long period of time. There are some cells that go into what's called G0, which is a resting phase, and they never divide again. A lot of neurons are that way. So just as a reminder, G1, this is where a lot of the growth of the cell occurs. It's where things like cellular respiration occurs. If it's a plant, photosynthesis is occurring. Internal structures are being used. Protein synthesis, which is transcription translation that we will be getting to. S, that's the DNA replication. We use the word synthesis to describe it. That's DNA replication, topic we just got through doing. And then G2 is really the kind of still more of the growth, but it's really set up to get ready for what's going to be happening next, and that would be the division of the nucleus and then the division of the actual cell. Okay, so prophase, first part of mitosis, or the M phase here. And what you end up having is that the nucleus is going to disappear. So the nucleus disappears. And the reason is that this mitosis piece, this really is the dividing of the nucleus. The nucleus needs to be divided first before the rest of the cell can be split, and then we get two identical daughter cells. So prophase, we end up having the chromosomes, they condense. So your DNA condenses. And what ends up happening when it condenses, just like when you see one strand of hair, it's really hard to see, but if you curl that up, you can see a lot of the hair. And so when DNA condenses, you actually start seeing it inside the nucleus, what would have been the nucleus. I'm just drawing it off to the side here, just drawing a couple of nuclei here off to the side. The other thing that happens is if it's an animal cell, the centrioles here, spindle fibers start forming and start stretching across the cell. And I'm just making them in dotted lines, but think of these are like tubes or strings that are being built, almost like a tower, if you will, but on that side, or a pipe that's just being built across the cell. Okay, each of these chromosomes has what's called a centromere. And that centromere, as these spindle fibers, is what they're called, spindle fibers. As the spindle fibers grow, they run in and attach to these centromeres. And that's what's occurring in prophase. Okay? The nucleus disappears, so this outer side nuclear envelope is gone. Some of the regulatory processes, some of the metabolic processes slow down or stop because the cell is really getting in the process of getting ready to divide. This is also the longest phase of mitosis. Interphase is the longest phase of the whole thing. By far, 90-95% of the time the cell is in interphase. It's only a very small fraction of the time that the cell is going to be doing the actual division of the nucleus, and my prophase being the longest part of that 4-step phase. So as the spindle fibers are formed, now remember in a animal cell it's called a centriole, but in a plant cell there's a cell wall here, and there is a centrosome section that you don't have to be aware of where the spindle fibers grow. So these centrioles are only in animal cells, not plant cells. All right, so as the spindle fibers grow and they attach to those centromeres on the chromosomes. Now these chromosomes, remember, were replicated here in the S phase. And so there's sister chromatids. Look in the notes to get that. Those centrochromatids are attached there at that centromere. And as things go by, as time goes by and the spindle fibers are formed, these chromosomes here that I just made, they're going to be pushed to the center. And again, because this is an animal cell, I'm going to continue to draw that. And those spindle fibers, as they push them to the center, they all start lining up. The chromosomes start lining up on the equator of the cell. So the chromosomes align along the equator of the cell. Okay. The spindle fibers are attaching to these centromeres, and it's all ready to go. The middle one. Okay, all the chromosomes are there in the middle. When you see these under a microscope, this is very, very diagnostic of what it looks like. The next thing is called anaphase. And anaphase is when the chromosomes start to come apart. And so we still have our centrioles here on the end. But what happens is the centromere, there's a part on it, it starts breaking down that spindle fiber. And it starts breaking down the spindle fibers in the direction that the spindle fibers grew. So that the spindle fibers then, basically, you can think of it forms kind of like a path by which the chromosomes travel. And the centromeres here have split, so that the sister chromatids are now split. And now we can call each of these sister chromatids a chromosome. And because they're moving back towards, it's the centromere that's moving back towards the centrioles to the opposite poles, the chromosomes have this distinctive V kind of shape to them. Okay? So here the chromosomes separate at the centromere. Okay. And then the spindle fibers, these spindle fibers start degrading, start breaking down, and it's the centromere that's doing that. It starts breaking it down so that the spindle fibers are where the chromosomes follow back to the centromere, or the centrioles, centrioles. So they're going back to those centrioles. And at this point, we still only have one cell, but now these are considered separate chromosomes, not sister chromatids, not just copies like they were back over here. Okay? The next part of mitosis, the last what's actually considered of mitosis in the upper level classes, that's called telophase. So our centriole that was over here, okay, the chromosomes have started moving and they're basically right back to the pole, and a nuclear envelope starts forming around them. Okay, so I'm just kind of drawing in some chromosomes. And now we start to get what's called a cleavage furrow happening. And that's what this is a cleavage furrow. And basically, the cytoplasm is starting to pinch in. That's the beginning of cytokinesis. So we have our nuclear membrane forming around each one of these. The centrioles are right outside. The organelles are starting to be divided up. This cleavage furrow kind of starts working its way in. If it was a plant cell, we'd have a cell plate follow in. The chromosomes begin to unwind. So the nuclear envelope or the nuclear membrane reforms. The chromosomes, they're so tightly wound, they're going to start unwinding a bit so that they're going to start disappearing. You're not going to be able to see them. The spindle fibers are gone. So those are gone because they're not needed anymore. They're just needed for mitosis. And a cleavage furrow forms. And this part is where we get the cytokinesis. Cyto meaning cell, and then the kinesis to split. And so what ends up happening is this cleavage furrow goes in farther and farther and farther. You end up getting two separate cells, two daughter cells that are exactly like the original parent cell that was all the way back over here in G1 at interphase. Each one of these daughter cells, and I'll just draw the cells here, and I'll just draw a nucleus just like that. Each one of those now goes back into interphase. Typically these are smaller than the original, and so they have to grow. So they're going to be in G1, and they're going to grow and do those processes where they basically get bigger and bigger and bigger. And once they get so big that the amount of fusion and some other things won't allow them to get much bigger anymore without the cell dying, they're going to divide again. Remember, this process is to help with growth of the cell so that we can get growth going on here, and then growth of the organism if it's a multicellular organism, just like you started off as a single cell and now you are bunches and bunches of cells. Okay? It also is for tissue repair, so that if, say, you get cut, then how do those skin cells get remade? It's through this process. This all happens in somatic cells. This is a very cursory piece, but it should help you figure out the parts of the cell cycle. Again, hopefully you have watched through and read through the notes section on this, and now you have this graphic organizer that will go into your lab book. That's all for now.